All right, now we go on to chapter 10, which is going to be going into the structure and function of DNA. So not just how DNA is assembled, but how DNA is copied, how DNA makes copies of itself, and how the information of DNA is used to actually be used to construct proteins. Now, at the beginning, they'd like to give you some examples of why molecular biology matters. So we've been spending time at the cellular level. Now we're going down to literally the molecular level. Well, probably one big one is the fact that all life on Earth shares the same genetic language, right? It's a universal genetic code. And that's why you see things like the ability to take DNA and insert genes, if you will, protein recipes into other organisms. Very good example of that being insulin. They can actually take the DNA for human insulin, it's a recipe, and put it into something like a modified yeast, and the yeast is not going to ask where it came from. It just knows to read the A's, G's, T's, and C's and produce a protein. So that's one way which we can use genetic information to produce things in ways that we weren't able to do before. Now, granted, given the modern day, talking about the Zika virus is going to be a little dated because this was 5 years ago, and of course now we have COVID-19 that's become the new threat to us. But your textbook is obviously going to be reflecting a somewhat older time. Anyway, so in 2015, a great number of babies were being born in Brazil with unusual severe damage to their central nervous system and sensory organs. If it was one or two babies, it would be one thing, but they noticed that it was a lot of them. And so often when you see a pattern of something appear somewhere, you have people like epidemiologists, you have people whose job it is to sort out why, is there a disease spreading, what's going on. And this incident in Brazil, they noticed this pattern that the affected babies had neurological problems, they grew slowly, they had difficult feeding, often they had to be tube fed, and they had joint and muscle problems. So obviously the health officials were like, what is causing this? They were able to find a link between these abnormalities and a new pathogen called the Zika virus. By the next year, the WHO, the World Health Organization, actually issued a worldwide health emergency. Now, it was first discovered to infect humans back in the 50s, and it can be transmitted to humans by one particular species of mosquito. To most healthy adults, it's not dangerous, but they found that developing babies actually were susceptible to damage being caused by the presence of this virus. Now, I think even today, health agencies don't have very many weapons against it. There's no vaccine, and medicines can only treat the symptoms. And so it's one of the things where there are viruses out there that we know about, but we don't have a silver bullet for. We don't have something like a vaccine to prevent infection. Now, any virus, including the Zika virus, are essentially a simple structure of nucleic acid. Viruses, you could almost argue, are not really properly alive, right? Things that are alive had all these characteristics, such as using energy. Viruses don't really do that. Viruses are essentially a little bit of genetic material in a protein package. And that's it. There's no, there's no mitochondria, none of the other mechanisms that you think of associated with cells. In fact, to even make copies of themselves, they essentially have to hijack a host cell to do it. They have the code to make a protein, but they don't have the machinery for it. So they infect a host cell, and they take over the cell's machinery to be able to make copies of their genetic material and make copies of their protein coat. We'll get into that a bit more later on in the chapter. But to combat any virus, Zika or COVID-19 or any of these, really requires understanding how life works at the molecular level, right? Because especially since you're combating essentially little packages of genetic material, well, how does this work? How does genetic material bring about the production of proteins? How is it that we can interfere with that production? And again, it's kind of amusing that we have, again, every few years teaching classes like this, there's always a new virus, that's emerging onto the Earth. So at this point, Zika may seem a little dated, but again, every edition of the book, unfortunately, there's a new big bad to deal with. All right. Now, DNA is a molecule, just as chemical stuff. It was known to be a part of cells by the late 1800s, right? So people who chemically broke cells down and identified literally the stuff inside, so lipids and all that. So DNA was known as a chemical, but Mendel, who's someone we'll be hearing about in the next later on in this section, talking about genetics, Mendel and other geneticists didn't know what DNA's role was in the business of inheritance. So it was known to be stuff, but they didn't know what the stuff's job was. By the early 1950s, a series of discoveries had convinced the scientific world that this was actually the molecule that acts as the hereditary material. So as the technology improved, being able to image things like molecules better, being better able to study these materials, and then that gave them the idea of, okay, we have hypotheses as to the role of this molecule, and eventually we were able to sort out that, yes, this is actually the hereditary material of cells. Now, at first, they knew that there was this stuff made of these nucleic acids, but the first thing that was a real challenge was, what was the three-dimensional arrangement of atoms? You might say nowadays, if you said, show me a picture of science stuff, people would draw a DNA molecule, right? We instantly think of this double helix. But back at, again, less than 100 years ago, what wasn't understood was, what was the shape of this molecule that allowed it to serve as a means of storing genetic information, being able to generate copies of this information, and to be able to pass it from generation to generation, either new cells being produced in an individual or new individuals being produced in reproduction. Now, a little bit of review from chapter 3. DNA and RNA are nucleic acids. They consist of long chains, otherwise known as polymers, of chemical units, monomers, called nucleotides. So a bunch of nucleotide monomers strung together form a polynucleotide, right? So that's what we call a nucleotide polymer. Polynucleotides can be insanely long. And again, the genetic alphabet is only 4 letters, right? The 4 different types of nucleotides, A, C, T, and G. So if you have a short alphabet, if you're going to have any number of words, the words wind up getting very long, right? There's only so many combinations if you have words, spelled with, say, 4 or 5 nucleotides. So to have the huge variety of proteins out there, it means that the code for these proteins, to have so many different proteins, means that they're going to get very long. Now, getting back to the chemistry, nucleotides are joined together by covalent bonds between the sugar of one nucleotide and the phosphate of the next. So in creating this polymer, you wind up with this repeating pattern of sugar, phosphate, forming that long strand. If you think of the 2 strands of the helix, the sides of that ladder, okay, that's the sugar-phosphate backbone. So the nucleotides are held together the long way by bonds between the sugars and the phosphates. They join to each other in the middle, right, the rungs of that ladder, by hydrogen bonds. We'll get to that a little bit more in a moment. So the nitrogen bases are arranged like ribs that project from the backbone, or, as I often say, like rungs, on a ladder. The bases are the part that's actually different from, let's say, DNA, nucleotide to DNA nucleotide. The sugar and the phosphate group are going to be the same. So if you're looking at DNA, you're going to have a deoxyribose and a phosphate group, deoxyribose phosphate group, right? That part that forms the backbone is the same. What's going to be different from nucleotide to nucleotide is that nitrogenous base. So again, in DNA, the sugar is called deoxyribose because compared to ribose, it's missing an oxygen, right? Deoxyribose. The full name for DNA is deoxyribonucleic acid. nucleic referring to DNA's location in the nucleus of a eukaryotic cell. And again, there's lots of different ways we can represent this visually, right? There's the classic double helix. If you unwind that, so you have two parallel strands with the bases in the middle. Okay, you can see that repeating blue and yellow in the diagram. That's referring to the sugar phosphate backbone, right? The fact that each one of those nucleotides has a sugar and a phosphate, and that's how they join to each other. And as you look through these diagrams, again, there's different ways of representing this. And again, just seeing it represented in different ways, it's kind of handy because some are really simple in terms of just showing like sticks that are short and long versus the diagram that's showing you the actual binding from the phosphate group to the sugar of the next. So again, it's always helpful to see different representations of these things, but to zero in on just one DNA nucleotide, right? Again, representing in yellow, you have the phosphate group. Represented in blue, you have the sugar, in this case deoxyribose, because it's a DNA nucleotide. And then here's the base, right? So that's the part that's going to be different. There's going to be 4 different bases. When you're looking at DNA polynucleotides, you're going to have A's, G's, T's, and C's. Here's a brief video.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides.
One difference between DNA and RNA is the type of sugar their nucleotides contain.
DNA contains the sugar deoxyribose,
while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugar are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so again, some of this is repeating from earlier in the semester, but the 4 DNA nucleotides differ in the nitrogenous bases, right? So the sugar and phosphate parts are the same, and again divided into 2 forms. Thymine and cytosine are single ring structures. Adenine and guanine are larger double ring structures. And again, if you're talking about RNA, you're substituting uracil for thymine. Also, the RNA sugar is ribose instead of deoxyribose. Moving on here. So again, just keep in mind that the major differences are single-stranded RNA versus double-stranded DNA. Substituting uracilforthymine. Now, if you've studied biology, sometimes you've heard of these two guys called Watson and Crick, James Watson and Francis Crick. These are the guys who determined that DNA, they knew was made of this nucleic acid stuff, but they didn't know the structure. These are the guys that got credit for determining it was a double helix. It was a helix with a uniform diameter made up of 2 polynucleotide strands. Now, what's finally making it into the textbooks in the modern era was these guys were working on this, but they're having trouble really determining the shape of the thing. They're visiting the laboratory of Maurice Wilkins in King's College in London. And I love this phrase. They saw an X-ray image of DNA produced by Wilkins' colleague Rosalind Franklin. To be more exact, they broke into her lab and looked at her stuff. She really did not tolerate people very gently. And so she was really not wanting to work with these guys, and they were like, well, we're going to see her images. So I love this. They saw an X-ray image. They saw it against her will, but anyway. So seeing that X-ray image, they then used that visual to be able to construct this double helix form. And they used wire models. This is a sort of classic image of the two of them looking at the way in which they sorted out the anatomy of this. And here is the less well-known Rosalind Franklin. Now, it's funny because years, going back decades, you never even heard of her. So at least they're giving some credit to her. They're still coming around to the point of saying that she was largely responsible for them having the visual, but we're getting there. All right. So, as you know, a DNA molecule is like a ladder twisted into a spiral, right? The sides of the sugar-phosphate backbones. Each rung is a pair of bases connected by hydrogen bonds. And the base pairing rules dictate that the side-by-side combinations, dictate the side-by-side combinations that form the rungs of the helix. And again, remember that they came in 2 different lengths, right? There were long ones and short ones. So you had to have pairing of long to short. If you had 2 longs, it would have widened that ladder, if you will. If they had 2 shorts, it would have narrowed, right? But equal distance. So that was one thing they determined was, okay, the pairings are pyrene to pyrimidine. All right. So next diagram coming up is just going to show, again, more visual ways of looking at this. So again, sometimes it uses shapes that emphasize the complementarity. Others show, more specifically what the hydrogen bonds look like. So again, just different ways of seeing this represented, right? You commonly see this ribbon model where you see the colors representing the sugar and the phosphate and the different bases. This is more of an atomic model showing the actual arrangement of the individual components. So not just blobs of sugar and phosphate, but you're actually seeing, okay, where is the sugar binding to the phosphate, etc. And then something that's more like a 3D model.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere.
The yellow and red phosphate groups make the 2 sugar-phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar-phosphate backbones.
Okay, so back to the history part. So in 1953, these guys made a big splash in the scientific world by, it was actually a fairly short paper. It's funny, you talk about Darwin and the monstrous book he produced. This is actually a fairly short paper proposing this molecular model for DNA. And 9 years later, in 1962, Watson, Crick, and Wilkins, the lab in which Rosalind Franklin worked, they received the Nobel Prize for their work. But unfortunately, the means by which Rosalind Franklin was able to visualize these molecules was by using X-ray photography. And she had died of multiple cancers in 1958 at a very early age. She was only 38, and was not eligible for the Nobel Prize, which you can only win that prize if you're a living scientist. So it's kind of sad that someone who is so critical to our understanding of the anatomy of DNA is for the longest time not even really mentioned. So at least she gets some credit, but unfortunately not the Nobel Prize. All right, so in that paper, they wrote that the structure they proposed immediately suggested a possible copying mechanism for the genetic material, right? Because part of what was the mystery was, how is it that this is duplicating? Right? How is this molecule constructed so that we can see how it makes copies of itself? So that double helix with the base pairing, that structure pointed towards a molecular explanation for life's unique properties in terms of reproduction and inheritance, right? Making copies and being able to pass copies of genetic material on to offspring. That arrangement of parts within the molecule affects how it works within the cell, right? So here we are back to another example of structure and function. Now, if you think about it, when a cell reproduces, even go back to something like an amoeba, right? A single-celled organism, or a bacterium, to make a new individual, right, you see that the cells sort of pinch into two new cells, but before it does that, it has to make a copy of its DNA so that the offspring cell gets one copy and the original cell keeps a copy. And that model of DNA suggests that each half of that helix, each DNA strand, can serve as a template or mold to direct how the other strand will be made. So again, you know about the base pairing business. Well, let's look at DNA replication. So again, replication means copying. Right? If you ever watched the Blade Runner movies, right, they talked about these duplicate human beings being called replicants. Right? So replication is making copies. If you see DNA replication, and again, this is something else that was worked out with research, but we now know that the parental molecule unwinds, and each half of the parental molecule gets a new strand formed as a complement to the original strand. At first they didn't know, was one copy brand new and one copy just the old one, but now we know it's what they call semi-conserved. It's the fact that each new strand is half the original strand and half a new daughter strand.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strands joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strand and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand, while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Okay, so ASE, remember that usually refers to an enzyme? So DNA polymerases are specifically the enzymes that make the DNA polymers, right? They're making those covalent bonds between the nucleotides of a new DNA strand, and they're also involved with repairing DNA that's damaged by things like toxic chemicals or high-energy radiation. So they not only make these strands, but they're also sort of like copy editors, right? They're there to look for mistakes. They're there to look for, if you will, typos and repair them. So DNA, again, it starts as it begins on a double helix at multiple sites. Think of it this way. Look at that big textbook you've got, or I guess look at the file you have if it's electronic. And if I said, okay, I need a copy of this. Now, if one person started at the beginning of the book and started making a copy from beginning to end, that would take a long time. But if multiple people started copying the book at different chapters, then those people working simultaneously could make a copy a lot faster, right? So the fact that it starts at multiple sites called origins of replication means that you get much more rapid copying than if it was just starting at one end of the strand and working all the way to the other end of the strand. So this copying ensures that the body cells in a multicellular organism all have the same genetic information. And again, it's also the means by which genetic information gets passed along to offspring. But if you think about that, every time your body makes a new cell, it's got to make a copy of that genetic material because all the cells have that same original genetic package. Think about making copies of the textbook without making a typo. Right? Mutations are random typos in the genetic code. So just the thought of the fact that constantly your cells are making new cells and the copies of the DNA are, for the most part, pretty exact. So another way of looking at this visually. So again, the original double helix is represented in blue strands, and the origin of there's multiple origins of replication, so it starts at multiple points, and eventually all those sections fuse together. So at the end, you have two daughter DNA molecules, half original parental strand and half new daughter strand. Now, the job of DNA, of course, is providing instructions to the cell and to the organism as a whole. A genotype is the genetic makeup of an organism. So that would be the actual heritable information coded in those nucleotide bases. The phenotype are the physical traits, right? So the genotype are the genes, the phenotype are the physical manifestations of those genes, right? The things that are coded for in those genes. And we'll get into the whole business of heredity in the next section. So, how do you get from DNA to a protein? Again, DNA is, think of that as nucleic acid language. RNA is also nucleic acid language, right? They're both languages of nucleotides. But proteins are amino acid language, right? These are chains of amino acids. So how do you get from the information on DNA to a strand of protein? 2 steps. The first is transcription, and this is the transfer of genetic information from DNA into an RNA molecule. Now, this is the same language, right? So if you're making a copy in the same language, we call that transcription. So if I'm speaking in English and you're writing your notes in English, you are transcribing what I'm saying. Well, if you took those notes and then converted them from English to Spanish, that would be translation, right? Now you're changing languages. So translation is transferring the information from RNA, which is ribonucleic, language, into a polypeptide, which is amino acid language. So translation is taking that information and changing it from one type of information to another. The function of a DNA gene is to dictate producing polypeptides. And so this business of genes and proteins really is one of information flow. So when you hear that phrase, the central dogma, what they're talking about is the flow of information from DNA to RNA to protein. So again, DNA is in the case of eukaryotes, is retained in the nucleus. The DNA does not leave the nucleus and go bouncing all around, bringing information to the ribosomes. And I always like to use the reference of, grandmother's cookbook, where grandma is not going to let you take the cookbook home, but she will let you make copies of recipes. Well, that's what's going on when you transcribe. You're making a copy of a sequence of DNA, a functional sequence called a gene. You're making a copy of that in the form of RNA. That RNA, messenger RNA, can now exit the nucleus. Right, so the DNA just rewinds back after you make that transcription. It's ready for making the next copy, right? So the DNA stays there in the nucleus, and copies of specific genes are sent out into the cytoplasm. Well, that messenger RNA exits the nucleus, goes into the cytoplasm, where it's read by a ribosome, and that is where that information encoded in the messenger RNA is translated by the transfer RNA. Right? So transfer RNA is transferring amino acids. It's also translating. At one end, it's holding an amino acid. At the other end, it's reading that messenger RNA. So again, transcription, you're making a copy in the same language. Both are nucleic acid language. When you change from nucleic acid language to protein language, that's translation. So again, this language of nucleic acids, we've had, talked about before. You've seen some videos of it before. It's just a linear sequence of nucleotide bases. A typical gene, because the alphabet's only 4 letters, is a few thousand nucleotides long. So when you reproduce these in your puzzles or your pop bead kits or whatever, obviously you're making something which is just like a few nucleotides long. To make a proper gene, you'd be popping together little beads all night long. When a segment of DNA is transcribed, the result is an RNA molecule. And again, this is transcription because the nucleic acid language of DNA has simply been rewritten, transcribed into a series of bases of RNA. So the sequence of nucleotides of the RNA molecule is going to dictate the sequence of amino acids of the polypeptide when it's read out at the ribosome. Now, thankfully for your sakes, the way in which this happened has been worked out already. The flow of information from gene to protein is based on a triplet code. So the instructions for the amino acid sequence of a polypeptide chain are written in DNA and RNA in a sequence of 3 base words called codons. So they're not read GCT, they're read, GCT or TAG, right? They're written in 3 base words called codons. And those 3 base codons in the DNA are transcribed into complementary, right, matching 3 base codons in the strand of RNA. Then those codons get translated into amino acids forming the polypeptide. So here's looking at it again. So again, starting with the blue DNA molecule, that unwinds, a complementary strand of RNA is formed, and that is going to be what exits the nucleus, goes out to the ribosome, and at the ribosome, transfer RNAs are going to be reading the codons at one end of the molecule, and if it matches, then, oh good, I have the amino acid that is going to be represented by those codons. So you're basically going, again, transcription, it's the same language. Then when you get to the ribosome, that RNA is going to be translated. So those codons, those groups of 3 bases, are going to get translated into individual amino acids. So remember that table you've seen, right? You read that table in groups of 3 bases. That's because a codon represents an amino acid. Now, the genetic code is a set of rules that convert a nucleotide sequence in RNA to an amino acid sequence. Now, of the 64 triplets, 61 code for amino acids, and 3 are stop codons, indicating the ribosomes, okay, that's the end of the polypeptide. So a given RNA triplet always specifies a given amino acid. So if we look to, again, another round representation of the code, starting at the center and moving outward. So the diagram you saw in the lab was starting at the outside and moving inward. This one starts at the center and moves outward. So again, these are all ways of representing the same information. In this way, you start at the center, and then when you get to the outermost ring, you will then be at the amino acid that is coded for by that particular triplet. So in this case, you start in the center and move your way outward. So in the case of the code AUG, right, A in the center, next ring out U, G in the outer ring, that codes for methionine, MET, that's the start codon. So again, just different ways of representing the same information. Now, as I said before, this genetic code is nearly universal. It's shared by organisms from the simplest prokaryotes to the most complex plants, animals, and fungi. This universality of the genetic vocabulary suggests that this arose early on, right? The things that are shared by the most living things probably developed earliest in the evolution of life. So the fact that you have this genetic vocabulary shared by everything suggests that this developed very early in the evolution of life and has been passed on over all these millennia to all the organisms found on Earth today. Now, because diverse organisms share a common genetic code, that's why we can program species to produce a protein from another species by just transplanting DNA. And so this ability to mix and match genes is now coming up in things like agriculture, medicine, research. In this case, you see this mouse showing some glowing proteins, right? If you put fluorescent light on this mouse pup, you can see that parts of it are glowing. And of course, the first question is, why are you making mice that glow? Well, it's not so much that you want to see the mice glowing. It's that the gene associated with this type of fluorescence in jellyfish can be used to label a gene you're transferring. So let's say you're transferring a gene that's involved with making a certain type of enzyme. It would be hard just looking at the rat pups formed, after this sort of genetic work, and just looking at them and saying, oh, did the transfer work? But if you take the gene for that enzyme you're following and attach that glowing protein from a jellyfish to it, if the transfer worked, you not only transferred the enzyme gene, you also transferred the gene for the glowing. And that's why we do this. It's a type of labeling. So it's not so much just for grins and giggles we can make a mouse that, that glows. It's that, okay, this gives us proof that we transfer the gene that was attached to that glowing protein. So it's used as an indicator. There's other uses for it, but this is a common way it's used in the laboratory. Okay, so transcription. Same language. You're transferring genetic information from DNA to RNA. So just like DNA replication, where you're making a copy of DNA, you've got to unwind those two DNA strands. And so just as you had those replication bubbles, right? You had those points where they're unwinding for the copying to take place. In this case, instead of the unwinding leading to two new strands of DNA, you're just going to be using one of those two strands to serve as a template for making that strand of RNA, right? So the other strand just, sits back and waits to be wound back up with the other strand. So only one serves as a template, because if you think about it, if you know what one half is, because of pairing rules, you know what the other half is, right? So only one strand is served as a template. Now, the nucleotides that make up that new RNA molecule take their place one at a time along the DNA template by forming hydrogen bonds with the nucleotide bases already there. So the same pairing rules, except for U rather than T pairs with A. And the enzyme that is doing this job now is RNA polymerase, right? Because it's making an RNA polymer. Now, the point at which this begins, right? So if you're making copies of the DNA, you've got multiple replication bubbles because you're copying the whole thing. How do you know where to start transcribing that DNA to form that RNA? Well, there's something called a promoter. It's the signal. It's a nucleotide sequence that is located in the DNA at the beginning of a gene, and it's the specific place where the RNA polymerase comes and attaches. This first phase of transcription is called initiation, right? If you join a club, there might be an initiation ceremony, right, where you join that club, your beginning of your association with those people. The first phase of transcription is initiation. It's initiating this production of an RNA strand. So it's the attachment of RNA polymerase to that promoter, the point that indicates the beginning of a gene, and that's where RNA synthesis will start. Now, during the second phase, elongation, that kind of tells you what's happening, right? that RNA strand is elongating. It's growing longer. And as it does so, that RNA strand peels away from the DNA template. So once that's peeled away, now those two DNA strands can wind back together again in the region that was already transcribed. The third phase of transcription is called termination. And you can kind of guess what that means. Termination means the end of something. So RNA polymerase, copy, makes that complementary strand. It reaches a special sequence of bases in the DNA template called a terminator. Right, so just as there was a code called a promoter, which says, here's where it starts, now you have a bit of code called the terminator saying, this is where it ends. So once it hits that terminator, the polymerase detaches from the RNA and the gene, and the DNA strands finish rewinding, and now you've got a strand of RNA. All right, so I'm just going to go through this stepwise. So again, here you've got the RNA polymerase, that's the big peach-colored blob there, and that's the enzyme that's matching RNA nucleotides to the complementary bases on the strand of DNA. So as the transcription proceeds, that strand of RNA peels away, right? It's not making a copy of DNA where they're going to, remain attached. This is something where it's going to peel away and the DNA is just going to rewind back together again once it's gone. So the steps are, again, beginning with the RNA polymerase joining where the promoter DNA is, right? That's initiation. Okay, as it works its way along one half of that DNA strand, matching those complementary bases, right? You have a growing RNA strand, that's elongation. And then, of course, termination is when it hits that code that says, okay, that's the end of the gene, that detaches, and now you've got this completed strand of RNA.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Okay, skipping over the seams there. Now, the interesting thing is when that is finished, right? When you finish having that transcription, okay, that in a prokaryote immediately functions as messenger RNA. So it is then translated into protein. But in a eukaryote, it's localizing transcription in the nucleus. And the RNA actually gets processed before it exits the nucleus into the cytoplasm for translation. And one kind of RNA processing is the addition of extra nucleotides to the ends of that transcript, at the end of that strand. These additions are referred to as the cap and tail, and it marks the messenger RNA for export from the nucleus and helps ribosomes recognize the RNA as messenger RNA, because keep in mind you've got rRNA, tRNA, and mRNA. I always think of it as putting on your hat and your shoes before you leave the house. But another type of processing is really interesting. There are known coding stretches of nucleotides, as I mentioned in the video earlier. So there's these stretches that don't code among the stretches that do code for amino acids. Most genes of plants and animals include such internal non-coding regions called introns. The coding regions, the parts of the gene that are actually expressed in polypeptides, are called exons. Now, both exons and introns are going to be transcribed from DNA into RNA. But before the RNA leaves the nucleus, the introns, the non-coding parts, are removed, and the exons are joined together to form an mRNA molecule with a continuous coding sequence. This whole business of cutting and pasting is called RNA splicing. And there's a lot of questions about this. Why even have these stretches that don't code, right? Well, they think RNA splicing has played a role in our development by allowing our roughly 21,000 genes produce several times that number of polypeptides. So in other words, the way in which that RNA strand is edited will influence what the polypeptide is it codes for. So by varying the exons that are included in the final messenger RNA means that you have different variations of codes that result from it. So it's interesting. You take out the ones that don't code. It's how the remaining exons are used that is thought to be part of how we've developed different types of polypeptides. We're not going to get into the deep of that here, but it's just kind of interesting. People have wondered, why do we even have that whole thing? So again, the DNA, which has both exons and introns, is transcribed, right? The whole thing is copied. The cap and the tail are added onto the messenger RNA. The introns get snipped out. The exons get sewn together. And now you've got a completed unit that can leave and go out into the cytoplasm. Now, translation, as I've said before, you're converting between languages, right? So like English to Spanish or Spanish to German. This is going from nucleic acid language to protein language. And so now you've got something a little more complicated than transcribing. Transcribing, you're just, in essence, making a copy. Well, now you've got a whole different ballgame here. The first important ingredient required for translation is that mRNA produced by transcription. Once that messenger RNA is present, the machinery used to translate mRNA requires various enzymes and sources of chemical energy, like ATP. It takes energy to carry out this process. In addition, translation requires ribosomes and transfer RNA, the RNA that's going to be bringing the amino acids represented by the codons. So translation of messenger RNA into the amino acid language of proteins requires an interpreter. And that's what transfer RNA is. And that's why I often think of it as the translator RNA. It's a molecular interpreter that's going to match amino acids to the appropriate codons to form the polypeptide. So two things have to be done by your transfer RNA molecules. It's got to pick up the appropriate amino acids, recognize the appropriate codons in the messenger RNA. So the unusual structure of this molecule, which you're going to see in a couple of slides, enables them to do both at the same time. So a transfer RNA molecule is a strand of RNA about 80 nucleotides long, but it's all twisted and folded onto itself. At one end is a special triplet of bases called an anticodon. This is complementary to the codon triplet on the messenger RNA. So messenger RNA is read in triplets, right? In these codons. Well, the complement to a codon is an anticodon. And so on the transfer RNA, you have the anticodon complementary to the codon on the messenger RNA. So during translation, the anticodon on the transfer RNA recognizes a particular codon on the messenger RNA by using those base pairing rules. At the other end, right, so you got one end with the anticodon, the nucleic acid language. At the other end is a site where a specific type of amino acid attaches. So even though all transfer RNA molecules look similar, there's going to be slightly different versions for each amino acid because they're going to be slightly different. They're going to be binding to slightly different amino acids. So here's sort of looking at it from a molecular point of view, where down at the bottom you can see the anticodon, and up at the top you see the amino acid attachment site. To the right is sort of a cartoon version that's a lot simpler to look at. So at one end is where the amino acid attaches, the other end is where the anticodon is. So where does the magic happen? at the ribosome. So ribosomes are the organelles in the cytoplasm that coordinate this functioning of messenger RNA and transfer RNA where the polypeptides are actually made. So ribosomes are made of 2 subunits. Each one is made of protein and a lot of the third type of RNA, ribosomal RNA. And that fully assembled ribosome has a binding site for messenger RNA on the small subunit and binding sites for transfer RNAs on the large subunit, right? So they are literally going to be bringing these 2 players together. So let me zoom in a little bit further. So again, we have these subunits. If you're using the kits at home, the puzzle kits at home, this is what that large piece of paper is representing. So you'll see these P sites, 8 sites, etc. on that diagram. So again, this is sort of a simple version, but you can see where the transfer RNA is going to park and it's going to be meeting up its anticodons to the codons of the messenger RNA that's going to be feeding through there, looking kind of like this. So the messenger RNA is being read through that ribosome, and as it moves through, complementary transfer RNAs are going to come in, matching its codons with their anticodons and bringing in amino acids to form that growing polypeptide. Now, conveniently, translation has the same 3 phase names as transcription: initiation, elongation, and termination. But instead of a initiation, elongation, and termination of a messenger RNA strand, you're having initiation, elongation, and termination of a polypeptide. So initiation is going to be bringing together that messenger RNA, the first amino acid carried by its attached tRNA, and those 2 halves of the ribosome. And again, that's where that cap and tail come in. It helps the messenger RNA bind to the ribosome. So you've got that cap, the beginning of the genetic message, and the end, showing the end of it. So initiation brings all the parties together. The messenger RNA binds to the small part of the ribosome. Initiator tRNA binds to the start codon, right? Remember there were codons that indicated the beginning and ending of a gene, and that's where translation is going to start. This large ribosomal subunit comes in and binds to the small one, so now you've got a functional unit here. All right, so as we see in this diagram, and again, this is something where animation is your friend, because showing this as a series of static pictures is not quite as helpful as seeing this in the form of animation. So go back to the animations that I've linked on YouTube, from YouTube to the Blackboard site. There are other ones out there, so you can browse around and see other ones, but this is one where I think animation really helps understand this. So initiation is, again, all these parts coming together. The messenger RNA, the transfer RNA with the MET amino acid, the small and then the large ribosomal subunits. So elongation is, again, just as you had elongation of the messenger RNA back in the nucleus, you have elongation of the polypeptide strand. Right, so the codons and the anticodons meet. Okay, if they meet together, then that means that code is representing a particular amino acid that is left behind as that transfer RNA leaves to get its next amino acid, and the next transfer RNA is going to be coming in with a new amino acid that binds to the progressively elongating strand. And again, this is something where I will merrily tell you to go look at animations versus me doing this verbally. The good part is when you do those puzzle kits at home, you're going to be reproducing this whole business using those cardboard puzzle pieces. So that'll give you a chance to put it all together, literally put it all together, and see how it works yourself. So elongation proceeds until the stop codon reaches that ribosome's A site. That completed polypeptide is freed, and that ribosome breaks apart into its subunits.
In translation, a cell reads an mRNA message and assembles the polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site, and the process is repeated.
One way in which
three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, what I'm going to do is scroll this back to this earlier part there. One way in which I remember these sites is A is arrival, P is when the peptide bond is formed. Let me zoom it up to that point. Wink. Okay, so A for arrival, P for peptide bond, E for exit. So if you're trying to remember these three spots along the ribosome, I found that was an easier way to recall it. Okay, so figure 10.9. I don't remember what page in your book it is. It is, I think it's 182. Nope, 183. Page 183 in your textbook. If you have that diagram down, it's really good representation of pretty much the whole works. It's showing the whole flow of genetic information from DNA to RNA to protein. Now again, in a eukaryotic cell, that transcription is happening in the nucleus, and the RNA is processed before it enters the cytoplasm. If you're looking at a prokaryote, they don't have a nucleus, right? So it's basically ready to roll as soon as it's transcribed. So a little bit of a difference there. So transcription and translation are the processes whereby cell genes are going to control the activities and structures of cells, right? It's making the proteins are what does all the work. So if you have this down, and I'm going to go through it stepwise in a second, if you have this down, you pretty much have a darn good handle on the whole thing. So again, transcription in eukaryotes is going to happen in the nucleus. You're transcribing a strand of messenger RNA from a DNA molecule. So again, you're using the same language. You're transcribing nucleic acid language to nucleic acid language. There's the whole business of processing, right? Snipping out the introns, sewing together the exons, cap and a tail being added. And now that messenger RNA molecule is ready to leave the nucleus. Meanwhile, out in the cytoplasm, okay, you've got transfer RNAs picking up amino acids. Pretty much everything that happens in a cell has got some enzyme involved, catalyzing a reaction. In this case, there are enzymes whose job it is to bring together an amino acid with its particular transfer RNA. And as with many things, this requires energy, right? So you're taking this amino acid and using energy to bind it to its particular transfer RNA. Well, once that messenger RNA leaves, it hooks up with the ribosome, as does the transfer RNA, and now you have the initiation of translation, right? You've got a transfer RNA bearing MET. It's going to bind with the messenger RNA. I'm moving on here. So initiation is where the parts all come together. Elongation is when progressive subsequent more transfer RNAs come in, bringing new amino acids to form peptide bonds in the P site. Then what happens is that strand is going to remain behind with the transfer RNA in the P site, and then that one that was previously holding the strand is going to exit, right, go off the E site. And so that keeps going until you get to the stop codon, at which point the whole thing comes apart. The transfer RNA, the parts of the ribosome separate, and now you've got that polypeptide strand.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the larger subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of the tRNA molecule has a set of three bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so that's how everything works now. Variation in genetic material occurs by something called a mutation. Any change in the nucleotide sequence of a cell's DNA is referred to as a mutation. These can involve large regions of a chromosome or just a single nucleotide pair, as you'll see in the example of sickle cell disease. Mutations within a gene are divided into two general categories: substitutions and insertions or deletions. So substitutions, the strand remains the same length. Insertions or deletions means that inserting, the strand is going to get longer, or deletions, the strand is going to get shorter. So you can imagine a simple substitution may not be as catastrophic as actually removing or deleting sections. And the example here being the molecular basis of sickle cell disease, where the normal hemoglobin DNA has, you'll see the base is CTT. So normally the complementary messenger RNA would be GAA, and that codes for glutamine and normal hemoglobin. In the mutant form, there's a substitution. You swap out that second thymine for an adenine. So you go to CAT. The messenger RNA, therefore, is GUA because you substitute, U for T. And now it codes for the amino acid valine. And so that one difference is enough to cause a difference in the shape of the protein and the function of that protein. Now, missense mutations involve a single nucleotide and change the amino acid coding, like we just saw in that last example. Nonsense mutations change an amino acid codon into a stop codon. Right? So missense means you're changing the amino acid coding. Nonsense mutations means that you stop it early in what would be the normal sequence. So that's why they refer to it as a nonsense mutation. It changes an amino acid codon into a stop codon, so it stops early. Mutations involving deletion or insertion of one or more nucleotides in a gene, called frame shift mutations, often have disastrous effects, right? Because when you're altering the length of the strand by adding or subtracting, you're going to be altering the triplet grouping of the genetic message past that point. So a frame shift mutation often produces a non-functioning polypeptide. So I'm just going to go through some examples. Let's start with base substitution. Right? So in this particular case, the first three codons are normal. Right? You see that they both code for methionine, lysine, and phenylalanine. But the fourth one, right, the fourth codon, you see that you're changing from serine, changing from glycine to serine, right? You're substituting that A for a G, right? So because if you go to that chart, right, and you're reading, that chart represents messenger RNA. So if you sit there and say, okay, here's the messenger RNA 1, 2, 3rd base, it's coding for a different amino acid. So this is a case where you're changing the actual sequence of amino acids. You can have substitutions that are silent. This would be an instance where you change a nucleic acid, change one of the nucleotides, but it still codes for the same amino acid. Because you notice in that diagram, there's many amino acids that have more than one possible codon representing them. So you can't have a silent mutation, right? Or it's a substitution where it still winds up coding for the same amino acid. Now, where things can get really rough is when you are talking about deletions and insertions. So in this case, you have the DNA nucleotides from the normal gene in blue, the mRNA nucleotides in red, and the amino acids that they represent in purple. So in this case, you're going to do is snip out one of the messenger RNA nucleotides. Well, now that you've done that, you've changed the reading frame, right? Instead of it just being one of those particular amino acids being different, right? So the first 3 are normal, the 4th is different, and then it goes back to being normal. Well, if you change the length of it, then that means that those reading of 3s is going to get messed up. So everything downstream of that deletion is most likely going to be wrong. So looking at this here again, so instead of it just being one amino acid that is different, well, now pretty much everything downstream. So whenever you snip one out or insert one in, you change the frames, right? You change the actual, everything, the reading frame basically referring to the group of 3 bases, right? The codon. Well, you're changing all the codons past that point if you're talking about deletions and insertions. So the next question would be, well, what even causes mutations? They can occur in a bunch of ways. Spontaneous ones can occur from just random errors. If you think about it, during DNA replication, copying the DNA, well, if you think about it, go back to thinking of making a copy of the textbook by yourself. Odds are you're going to have a typo in there. Right? So sometimes there are just random errors in the process of copying DNA rather, or in recombination. We'll get into that a little bit later on. Other sources of mutation are physical and chemical agents called mutagens, right? Gen referring to creating something, right? So a mutagen generates mutations. A pathogen generates disease, right? Pathology. The most common physical mutagen is high-energy radiation, right? So if you go to the, past the visible spectrum, you've got things like X-rays and ultraviolet light. These are things that can damage genetic material. The chemical mutagens come in all sorts of types. But because many mutagens, things that change the DNA sequence, can act as carcinogens, things that can generate cancer, you should avoid them as much as possible. Now, although mutations can be harmful, they can also be beneficial, both in the natural world and in the laboratory. One of the reasons there is such a diversity of genes in the living world, the example in one of the videos was talking about eye color, right? That's just one tiny example. But the diversity of genes out there all comes from mutation, right? It's a source of the diversity that we see in the living world. And because you have diversity in form, then what? If you have different forms, then there's evolution by natural selection being possible, right? If everything was genetically identical, then natural selection, as far as, in essence, choosing the best genetic setup, wouldn't be taking place. So if you look at some of the variety out there, things like just these tropical fish, the patterns and the colors, okay, all of that variation comes from mutations in individuals. Now, on to the viruses. A virus is an infectious particle consisting of basically genetic material in a box. A little nucleic acid wrapped in a protein coat, and sometimes even a little envelope of membrane. I'll explain that in a moment. And again, to me, viruses are sort of borderline, life, because they share some of the characteristics of living things. They have genetic material, they're organized, packaged in a, complex structure, but it's not made of cells. And it cannot reproduce on its own. So it's sort of a weird thing in the scheme of talking about living things. Now, the viral genome can be DNA or RNA, and it can be single or double-stranded. Right? And here's sort of your classic adenovirus. So inside that protein coat, the protein represented by these little gray spheres, if you sort of snip a little hole in there and look inside, there's DNA, and that's it. That's all the virus is. It's genetic material in a package. That's why something like an antibiotic that would act on, say, a ribosome isn't going to affect a virus, right? There's no ribosomes to interfere with. So viruses don't care. Viruses is like, yeah, that's nice, and just move on along. Something that affects a cell membrane isn't going to touch a virus because there's no cell membrane. All right, so the first viruses studied were viruses that attack bacteria. And they have the great name bacteriophages. Phage refers to eating. So the full name is bacteriophages, but often in books and research you hear them just called phages for short. A phage is a molecule of DNA enclosed within this package of proteins. And again, they can't reproduce themselves. What they do is they use other cells. They use host cells, in this case bacterial cells, to carry this out. Once these phages infect a bacterium, most of them will enter a reproductive cycle called the lytic cycle. Right? Some of them are also going to reproduce by an alternative route, the lysogenic cycle, in which viral DNA replication occurs without producing new phages or killing the cell itself. So here we see a photomicrograph. Okay, colorized transmission electron micrograph, 225,000x. Because again, viruses are tiny. You're seeing this sort of brownish zone here. That's a bacterial cell. If you're looking at this on a computer screen, that bacterial cell would probably extend well past the ends of your monitor. All right, so you think of how small a bacterial cell was. It's huge compared to viruses. Right, so you see in this illustration is 3 little bacteriophages in a row. The first one is sort of, you see this sort of empty part up here, but if you look down here, you could see the DNA being injected into that bacterial cell. So what happens is that in the case of bacteriophages, the DNA is packaged up in the head of the thing. It will use these proteins to, in essence, bind to the host cell, and then its DNA is injected into it. Once that happens, then this protein packaging just peels away. So, next diagram, 1025, is going to illustrate the 2 kinds of cycles for a phage named lambda that is known to infect E. coli bacteria. All right, so you can see that there's 2 pathways here. One is, as you see in the center of the diagram here, lytic. The other is lysogenic. Now, lytic is basically a pretty direct route. The phage attaches to the bacterial cell, injects its DNA, right? And here's the existing bacterial chromosome in a loop. That phage DNA circularizes and, in essence, destroys the bacteria's own DNA. So now you've got code to make what? More DNA, more protein. So now it's hijacking the ribosomes and other machinery of this bacterium to make new phage DNA, make new proteins. Those phages assemble, and then the cell lyses, releasing the phages. Right, so now these new phages can go and infect another cell. So this is pretty direct, right? Phages dump their genetic material into the bacterium, make copies of themselves, blow the cell open, and now you've got phages spreading elsewhere. But there's an alternative route. Okay, once again, the phage attaches to the bacterium, injects its genetic material, but instead of taking out the host DNA, the phage DNA grafts itself into the bacterial chromosome. We refer to this now as a prophage. So now you've got that viral DNA as being part of the bacteria's DNA. So instead of taking out the bacterial DNA and just making copies of itself, it becomes part of the genome of the bacterium. So now every time that bacterium duplicates its genetic material and divides, along with it are copies of the prophage. So each normal cell division, you're having more and more bacterial cells with that prophage being in there. Eventually, the prophage may leave the chromosome, and it will then go into the lytic cycle. So go back a couple of slides here. So in the lysogenic cycle, basically, you have these bacteria reproducing normally, but now you have all these bacteria with copies of that phage protein as opposed to just a phage DNA as opposed to a single one. Well, eventually, occasionally, a prophage will leave that bacterial chromosome and then it will go into the lytic cycle. So lysogenic cycle is just making lots of bacteria, each containing copies of that prophage. Occasionally, some will then go into the lytic cycle. Now you've got phages that will be released and infecting other cells.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages, which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
Okay, so now we often think of viruses in terms of animal cells, but there are also plant viruses. And viruses that infect plant cells can actually do things like stunt plant growth. And in the case of agricultural plants, diminish crop yields. Now, most of the known plant viruses have RNA as opposed to DNA as their genetic material. Many of them, like the tobacco mosaic virus, are rod-shaped with a spiral arrangement of proteins surrounding the nucleic acid. Now, there's no cure for most viral plant diseases, because again, you think of things like antibiotics, they're not going to touch viruses because they don't have the machinery that antibiotics usually act on. The big problem nowadays is that a lot of the times in these large agricultural businesses, something like harvesting machines that go through fields and pull plants apart and separate the harvested material from the rest, these are often moving from farm to farm to farm. So if one farm gets a viral infection, the chances are that that machine can then take particles of that virus that are trapped among the parts of the machinery to the next farm and spread the viruses there, and then to the next farm. So one big challenge is just preventing infection in the first place. But in more recent years, they're trying to do is breed or genetically engineer types of crop plants that are resistant to viral infection. So just an example of the tobacco mosaic virus. And you can see that the virus, because it's taking over cells, right? It's diverting cells from what they would normally be doing to make copies of itself. So now the cells are not, in this case, you see there's not as much photosynthetic material here. So without the photosynthesis, it doesn't have the raw material and the energy to do what it normally would be doing. So it can really be an issue with crops. There's a closer picture. Now, viruses that infect animal cells are common causes of disease, right? No virus is a greater health threat to our species than the influenza virus, right, that we call flu viruses. Like many of the animal viruses, it has an additional little trick. It's an outer envelope made of phospholipid membrane with projecting spikes of protein. What some of these viruses do, and you might say, well, wait a second, viruses don't have cell membranes. True. But what some of these viruses do is when they're leaving the host cell, they actually steal a little bit of the membrane and wrap it around themselves. Right? So as it leaves, there's a little bubble of membrane around it. One handy thing about this is the immune system in animals is relying on identifiers that are found on the membrane of cells, right? If it's coming up to a cell that is a cell from its own body, there are markers on that membrane that are like, oh, okay, you're on my team, and it doesn't attack them. So some of these viruses will steal a bit of membrane and allows them to sort of hide from the immune system within cells. So this is one virus that does have that outer envelope made of phospholipid membrane, and then little spikes of protein project through that to be able to bind to the next host cell. The other cool thing about the envelope is that instead of the proteins bind it to the next cell, but the envelope can allow it to merge that membrane with that of a host cell and dump the contents inside. So pretty nifty little trick there. Now, many viruses, like those with influenza, common cold, measles, mumps, AIDS, and polio, have RNA as the genetic material. Diseases associated with viruses with DNA include hepatitis, chickenpox, and herpes infections. And here's an example of a membranous envelope and enveloped virus. Now this next diagram is going to go through the reproductive cycle of a typical enveloped RNA virus, which is, in this case, the mumps virus. So just sort of zooming in, you see on the lower right hand is actual little mumps viruses. Here is each of these little guys. But let's look at the cycle itself. I'm going to zoom all the way in there. So first off, protein spikes enable it to bind to the host cell. That membrane it stole from another cell now merges with the envelope membrane merges with the cell membrane of the new host cell, and now the contents just get dumped inside. Once inside, okay, now the protein coat breaks down, revealing in this case an RNA strand. The viral RNA strand now is going to do is synthesize copies of itself. Okay, so what's the information in that RNA? Code for more RNA and code for the proteins that will make the package. So it synthesizes these parts, they assemble, and this guy exits, taking some of the membrane of that host cell with it. So again, you can see here these little mumps viruses. Kind of tricky to see that little protein spikes there, but yes, it packages in this envelope, which enables it to enter and exit host cells.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell, and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cells to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Now, not all animal viruses reproduce out in the cytoplasm. The herpes viruses, which are associated with chickenpox, shingles, cold sores, and genital herpes, are enveloped DNA viruses that reproduce in the host cell nucleus and get their envelopes from the cell's nuclear membrane. And copies of herpes virus DNA usually remain behind in the nuclei of certain nerve cells, where they can remain latent, where they can basically sit there and not do anything for long periods of time. And that's why some people who have things like cold sores, when they're under stress, suddenly the virus will start acting up, right? Well, it's because copies have remained inside, and when the immune system is challenged or various forms of stress, they become active and they start causing problems again. Now, the amount of damage a virus causes to the body depends partly on how quickly the immune system responds to fight the infection, and partially on the ability of the infected tissue to repair itself. So think of your classic rhinovirus, right? Your cold viruses. Usually you recover very quickly because you're constantly replacing those tissues lining the respiratory tract. You're constantly shedding off those cells and replacing them. But in contrast, the poliovirus attacks nerve cells, and nerve cells are typically not replaced. So the damage done to cells by polio is permanent. So in these cases, the only medical option is to prevent getting the disease in the first place with vaccines. Okay, so process of science, I'll give you an example here. Can DNA and RNA vaccines protect against viruses? West Nile virus first arrived in the US in 1999, and most humans who get the virus don't become ill. But in some cases, the virus causes a potentially fatal swelling of the central nervous system. Now, this is another virus spread by mosquitoes. There is no vaccine for humans. There is a vaccine for horses. But there's no vaccine for humans and no cure for those who become ill. So this is a case where, okay, you really want to find a way of dealing with this. Well, researchers tested an RNA vaccine containing a gene from that virus using cats and dogs. The cats are given high doses of the vaccine, low doses of the vaccine, or placebos, right? So something that is similar to the vaccine but not actually functional as the vaccine. All animals got booster vaccines 28 days after that first dose. After that time, they then exposed these animals to mosquitoes known to carry the virus and then see if they became infected, right? So if they became infected, then the vaccine did not protect them. So in the next diagram, you're going to see no cat infections at high vaccine doses and no dog infections at low vaccine doses. Right, so here's your test protocol. So they divide the animals into 5 groups. All were exposed to mosquitoes carrying the virus. They test the blood for presence of the virus and see, at what rate these animals were infected. So if you look at that table at the bottom, it's like placebo, okay, high number of cats are infected because obviously there's no vaccine there. The low vaccine dose, a fair number were infected. High vaccine dose, none were infected. But with the dogs, yes, you had a high rate of infection with placebo, but at low vaccine doses, it was very effective. So vaccines are operate in different ways in different animals. This is why you do animal testing, but you can't assume that something that works in one type of animal works in another. So, HIV. HIV is the immunodeficiency virus. It's associated with the disorder AIDS, acquired immunodeficiency syndrome. And this is an RNA virus with some nasty twists. It's a retrovirus. Now, usually people think retro, it's like, oh yeah, like clothing from the 70s. Yeah, not quite. Retroviruses have an unusual trick to them. HIV is a retrovirus, which is an RNA virus that reproduces by means of a DNA molecule. They call them retroviruses because they go backwards. They reverse the usual DNA to RNA flow of genetic material, of genetic information. These guys are RNA viruses that have an enzyme called reverse transcriptase. It's reversing transcription. Instead of transcribing DNA to make an RNA molecule, you're taking an RNA template to make a DNA molecule. So it's transcribing in reverse from the usual pattern. So these guys, again, this is another enveloped virus. Here's that protein coat. And here you have RNA strands and molecules of this reverse transcriptase, because this is something you don't find in every host cell out there. So this virus, the red dots are just showing examples of HIV on a white blood cell. Let me just zoom into the diagram itself. Okay, so this enters the cell. You've got the viral RNA. You've got the reverse transcriptase. This enzyme is now transcribing a strand of DNA from an RNA template. Okay, from that strand, you can then what? You can make the complementary strand. Now you have double-stranded DNA. What these sometimes do is they actually enter into the nucleus and merge with the host's DNA. So as you had prophages back with the bacteria, you have proviruses. They merge their DNA with that of the host DNA, and under circumstances not totally understood yet, at some point, sometimes that DNA will be transcribed into RNA. And you'll have these copies of RNA plus proteins coded for by the genetic material, forming new viruses, which can then leave taking a little envelope with them.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Okay, so how does HIV work? Well, it infects and eventually kills certain types of white blood cells that are part of your immune system. Now, there is not yet a cure for AIDS, but its progression, its ability to make copies of itself, can be slowed down by two categories of anti-HIV drugs. Both of these interfere with the reproduction of the virus, right? So if you can interfere with its reproduction, then you can interfere with its spread. The fewer copies of virus in the body, the less damage being done to the immune system of the host. Now, the first type inhibits the action of enzymes called proteases, which help produce the final version of HIV proteins. Well, without the packaging, the protein can't leave and infect other cells. The second type involves a drug called AZT, which inhibits the action of the HIV enzyme reverse transcriptase. So again, without that critical enzyme, the virus can't copy itself. Also, sort of a cruel little trick. Thymine as a nucleotide, base. What AZT basically is putting in fake thymine, for want of a better word. It looks like thymine, it binds like thymine, but it is not the same. Look at the two sugars here. So what happens is the virus is trying to make copies of its genetic material, and it's using fake thymine, right? And so what happens is it's knitting those into the genetic material, but it's not going to be properly functional. Now, many people who have HIV take a drug cocktail, right? And this is containing reverse transcriptase inhibitors and protease inhibitors. It's a mix. And that seems to so far be more effective than just giving the transcriptase inhibitors or just giving the protease inhibitors. So the two seem to be more effective at keeping the virus at bay and extending a patient's life. In fact, the death rate from HIV infection can be lowered by 80% with proper treatment, in essence turning it into a chronic disorder. And not a necessarily lethal one. But again, even in combination, you're not getting rid of all the virus. You're just keeping it at bay. You're keeping it from spreading. You're keeping it from damaging, the immune system. So because there's no cure yet, and if infected, at best you can sort of keep it, slow it down from causing damage. Prevention is still the best way of avoiding, dealing with HIV. So avoiding unprotected sex, avoiding needle sharing, things like that. Now, you thought viruses are weird? Let's talk about prions. Prions don't have genetic material. A prion is an infectious protein that is associated with brain diseases in several animal species. So while a virus has DNA or RNA, a prion is just a misfolded brain protein. Not genetic. It's just a messed up protein, right? It's a misfolded protein. And what happens is when a prion gets into a cell containing normal forms of the protein, this prion somehow converts normal protein molecules into misfolded versions. Those misfolded versions then clump together, and because we're talking about nerve cells here, it winds up disrupting brain functions. And again, we're still coming to understand how the heck this works, and because we don't totally understand how it works, we have not yet come with a good way of fighting the infection. So new viruses that suddenly come to the attention of medical scientists such as COVID-19 are called emerging viruses. Examples were Zika and West Nile virus earlier in the chapter. They'd been around there. They weren't totally new. They weren't novel like COVID-19, but they'd been around, but then suddenly became a bigger threat. Why? Well, there's three ways in which a virus suddenly becomes a big deal in the human population. One is existing viruses mutate. Right? Mutations occur in viruses just like they do, other things with genetic material. Or a new viral disease arises from the spread of an existing virus from one host species to another. So when you hear of things like avian flus or swine flus, these are things that are jumping to human hosts instead of their previous normal host. Or a viral disease that has been isolated from people can spread, leading to an epidemic. So something that was localized in small areas can be dispersed because we're now a very mobile organism on the face of the earth. So here's just a sample of some of the emerging virus outbreaks over the last century. Right, so things like Zika and AIDS, various H1N1 influenza, the West Nile virus, SARS, which is severe acute respiratory syndrome, and avian flu, and of course Ebola, right? So they're out there, and we have to learn how to deal with them. And a Nobel Prize winner named Joshua Ledberg warned, "We live in evolutionary competition with microbes. There is no guarantee that we will be the survivors. So if we're going to deal with emerging viruses, we have to keep working on the science, right? We need to keep understanding molecular biology and understanding evolutionary processes that lead to such things." And that's the end of the chapter.